The topic of ancient colonization in Europe is a subject of intense debate, with much discussion surrounding the samples obtained. These samples reveal significant levels of diversity, although this diversity diminishes as the Neanderthal morphological pattern emerges. In this video, several important questions need addressing. Firstly, did the variation arise from multiple instances of bottlenecking, hybridization, or a combination of both? Secondly, what was the extent of these events? Were they widespread on a global scale or more localized? The drastic climate changes likely played a role in shaping these scenarios, leading to population isolation, extinction of certain groups, and the subsequent hybridization of remaining populations. Over a span of 200,000 years or more, these climatic shifts potentially caused the demise of multiple groups. Concurrently, hybridization between the surviving groups and the influx of new migratory movements into Europe could explain the observed physical similarities. Altamira man, one of the most intact and well-preserved Neanderthal skeletons ever discovered, remains inaccessible at the bottom of a sinkhole near the town of Altamira in southern Italy. Over 130,000 years ago, he collapsed and succumbed to starvation in that very location. A human skull, encased in a thick layer of calcite formations, was found by cave explorers in 1993. The remains were situated in a cast borehole where stalactite growth was facilitated by water and limestone. Scientists refrained from extracting the bones for 2 decades to avoid causing irreparable damage. Consequently, archaeologists could only speculate about the individual known as the Altamira man. However, subsequent findings have surpassed expectations, providing valuable insights into this ancient cave dweller. The demise of the Neanderthals was a harsh one. Experts believe Altamira man perished hundreds of thousands of years ago when he became trapped in a natural well. Dehydration or malnutrition likely contributed to his death. Regardless of these circumstances, the cave system where he fell preserved his final resting place in a unique geological manner. Over time, his skeletal remains fused with calcite-rich stalactites, resulting in his current state embedded in layers of limestone. The estimated age of the remains ranges from 40,000 to 200,000 years. Due to the precarious position of the bones, only the head and a portion of one shoulder were exposed, while the rest remained untouched. Until recently, researchers had limited information about the Altamira man. By using uranium-thorium dating, they determined that the stalactite containing his head formed between 187,000 and 128,000 years ago. Remarkably, Altamira man now represents the oldest Neanderthal bone sample from which DNA has been successfully extracted. In light of these discoveries, what lies ahead for those seeking to unravel the mystery surrounding Altamira man? Researchers plan to run trials to check whether the DNA test can be sequenced. If fruitful, this sequencing could give significant new bits of knowledge with respect to Neanderthals and their job in human advancement. These discoveries and their potential future applications are incredibly promising for researchers who have struggled with the limited accessibility of the Neanderthal site. To reach the man trapped indefinitely within the rigid social hierarchy, you must embark on a claustrophobic descent through narrow crevices that take approximately 20 minutes. The inaccessibility of the location presents certain advantages. The original shaft through which he fell is no longer visible, as it has been filled with silt. Archaeologists are confident that the complete skeleton remains intact. In addition to obtaining DNA data, researchers have gained unique insights into the Altamira man's oral condition. They discovered that he was a young adult who exhibited signs of a dental ailment. Unlike other Neanderthal specimens, who would generally boast excellent dental health and a full set of teeth, Altamira man had calcified dental plaque deposits and 2 missing teeth. Some scientists speculate that future technological advancements will allow them to go beyond conducting in situ analysis of such artifacts. If the Altamira man were to be securely extracted, he could offer a wealth of new knowledge about an important brand of our ancestral tree. A recent project titled Virtual Excavation and Analysis of the Early Neanderthal Cranium from Altamira has produced a 3-dimensional representation of the skull. The discovery of complete Neanderthal skeletons is said to be an extremely rare occurrence. The morphology of the Altamira cranium aligns with the variability observed in Neanderthals, despite possessing traits found in older European samples. Some of these characteristics, previously unseen in Homo heidelbergensis specimens dating back 300,000 to 40,000 years, suggest that the archaic traits of Altamira may have emerged due to the physical isolation of early Neanderthal groups from southern Italy. Initially, the Altamira skeleton was thought to belong to Homo heidelbergensis until its DNA analysis confirmed it as a Neanderthal. Similar assignment strategies may be employed for future specimens. For instance, the Italian soprano fossil, dating back 400,000 years, holds significance in understanding the development of Homo neanderthalensis. According to archaeologists, soprano, an ancient find, is estimated to be around 450,000 years old. This places it right in the heart of the Middle Pleistocene era. The estimation is based on a careful examination of its geological, biological, and archaeological connections to a nearby Acheulean site known to be around 487,000 years old. Due to the skull's similarity to those of Homo erectus and Homo heidelbergensis, two extinct human species that existed in Europe before Homo neanderthalensis, scholars have taken notice. Many investigations have been made to clarify its origins. Although one study suggests a connection with Homo neanderthalensis, recent discoveries challenge the previous understanding, indicating that the skull is actually younger than initially estimated. Radiocarbon dating places its age between approximately 430,000 to 385,000 years ago. Consequently, it is suggested that soprano is present in European Middle Pleistocene fossils. Soprano's distinctive combination of archaic and derived characteristics presents a perplexing picture of European human evolution. The Middle Pleistocene era likely witnessed significant phonetic diversity, particularly during the time span of 1 million to half a million years ago, when a new form of humanity supposedly emerged and spread throughout Africa and Eurasia. Homo heidelbergensis, being a polymorphic and wide-ranging taxon, played a significant role in this argument. Throughout the Middle Pleistocene, numerous ancient human lineages associated with Homo heidelbergensis were identified, facilitating the recognition of regional variations or subspecies. Soprano, therefore, serves as the most suitable descriptor of the cranial morphology of the unidentified precursor variation of the last common ancestor species. In 1908, a mandible discovered near Heidelberg in Germany received the same Homo heidelbergensis. This mandible, dating back 600,000 years, held the distinction of being Europe's oldest hominid fossil for almost a century. However, during that time, no other Homo heidelbergensis fossils were found. For 70 years, the species was maintained based on the classification of around 20 Middle Pleistocene specimens from Europe, Asia, and Africa. These specimens exhibited a large brain and advanced tools similar to those of Homo erectus. Nevertheless, the revival of Homo heidelbergensis as a distinct species was unwarranted. Consequently, the Middle Pleistocene European specimens could tentatively be grouped with 2 or more Neanderthal clades, leading to the removal of Homo heidelbergensis from the hominin tree. Among the most fascinating aspects of this evolutionary tree are the recent genetic revelations and peculiarities. Initially, geneticists associated language with the FOXP2 gene, dispelling the misconception that Neanderthals lacked the ability to speak and communicate. The sequencing of the Neanderthal Y chromosome has presented more questions than answers. At first, scientists had the belief that Neanderthals and humans split apart around 590,000 years ago, based on a particular sample. But DNA tests showed that the Neanderthal Y chromosome had been completely replaced by the Y chromosome of a group from which Homo sapiens emerged between 150,000 and 350,000 years ago. There may have been a mating between Neanderthals and modern humans more than 100,000 years ago. As a result, the modern human Y gene was passed on to Neanderthals because it had useful traits. It is fascinating that the Neanderthal Y chromosome is more firmly connected to current people than to Denisovans, who lived in Eurasia simultaneously as Neanderthals. This contradicts the nuclear genome, which clearly indicates that Neanderthals and Denisovans share a sibling lineage separate from modern humans. The latest comprehensive data on the Denisovan and Neanderthal Y chromosomes suggests that earlier Neanderthals likely possessed a Y chromosome similar to Denisovans. The Neanderthals nonetheless acquired the advanced human Y chromosome plane in the wake of interbreeding with modern people between 370,000 and 100,000 years ago. Neanderthals were discovered in different locations of Eurasia, separated by tens of thousands of years, and their Y chromosomes were similar to those of modern humans. However, only a small portion of the Neanderthal genome is identical to modern human DNA, and the Neanderthal Y chromosomes were repeatedly changed. To investigate further, the researchers studied DNA from 3 Neanderthal bones dating back fewer than 53,000 years and 2 Denisovans. Various methods were used to extract hominid DNA, and specific probes were developed to target the Y chromosomal sequences. These sequences were then compared to those of modern individuals from Africa and other regions. The researchers sequenced approximately 7 million base pairs of each archaic individual's Y chromosome. They discovered that the Y chromosomes of all three Neanderthals were more closely connected to modern humans than to Denisovans. This pattern, while unexpected, has been documented in earlier investigations. Besides, the mitochondrial genome of Neanderthals is more like that of current people than that of their nearby cousins. Both the Y chromosome and mitochondrial DNA provide consistent evidence, revealing a remarkable story. Scientists found that an early population of Neanderthals had mitochondrial DNA resembling that of Denisovans. However, a group of early modern humans who migrated from Africa to Eurasia and interbred with Neanderthals eventually replaced the Denisovan-like mitochondrial genome. Human evolution remains a complex puzzle with the many missing pieces. Nonetheless, the discovery of additional fossils and their genetic information is gradually unraveling the mysteries. The study of ancient DNA has offered information on the links between contemporary humans, Neanderthals, and Denisovans. Denisovans in Asia and Neanderthals in Europe are thought to have been separated from Homo sapiens around 700,000 years ago. The discovery of the earliest Neanderthal specimen, dating back nearly 400,000 years, in a Spanish cave produced shocking conclusions concerning their genetic makeup. Mitochondrial DNA analysis indicated a closer resemblance to the Denisovans rather than other Neanderthals. This suggests that late interbreeding with modern humans led to the complete replacement of Neanderthal mitochondrial DNA. Paleogeneticists have theorized that the Y chromosome of Neanderthals was entirely replaced between 150,000 and 350,000 years ago. The introduction of a modern Y chromosome into the Neanderthal gene pool played a crucial role in increasing genetic diversity and aiding their survival. However, paleoanthropologists approach the topic cautiously, considering the concept of the population rather than distinct species when determining the source of the new Y chromosome. Around 350,000 years ago, both Europe and Africa were uninhabited. The hereditary likeness between the Neanderthal Y chromosome and that of present-day people suggests that it began from Homo sapiens-related populaces. Researchers proposed two expected situations. Modern people moved out of Africa and interbred with Neanderthals in Europe, or there was a populace on the two land masses before this blending. Unfortunately, our current knowledge cannot definitely address this issue, leaving the question of the mysterious male progenitor of humans and Neanderthals unanswered. The tale of human evolution continues to be revealed, challenging our preconceptions and deepening our grasp of our intricate shared history. This understanding is being aided by continuous genetic research and archaeological findings that throw light on our distant ancestors.